Hello and welcome to another Conversation with Giants in Medicine. I'm Ushma Neal with the Journal of Clinical Investigation, and today Dr. John Oates of Vanderbilt University joins me to discuss launching the field of clinical pharmacology and his research on the metabolism and biosynthesis of eicosanoids. Oates continues to be active in the division of clinical pharmacology that he founded 50 years ago, and wherein he made seminal contributions to our understanding of how the human body processes drugs. So welcome, and thank you for joining me today. I would love to start by hearing about where you were raised and your parents.
I was raised in Fayetteville, North Carolina, which at that time was a small town, about 35,000. My parents, my mother started out as a school teacher before she got married. And my father began his career as a school teacher also in a one-room schoolhouse. And he later became a lawyer and also a historian. So that was the environment that I grew up in. They both had interesting libraries in the home. My mother was attentive to the fact that I might like to read something different from what they read, so she found books that were more appropriate to a boy growing up. He lived about 3 blocks from the library, the public library, that I could walk to and pick out what I wanted to read, which I think was important, rather than getting books delivered to me or chosen for me. They had the shelves in the library organized so that by height, so the books were interesting for small children would be on the bottom shelves, and as you grew up, your level of books would change. One set of books that my mother got for me was about 10 volumes called The Book of Knowledge. That was sort of like an encyclopedia, but it wrote about subjects in a more interesting way. And a lot of science, geography, history in it. And I think I read most of that progressively as I was growing up. It was an interesting story in the family about how my mother bought these. It was during the Depression, and my father had a strict policy that nothing would be bought on credit or in the installment plan where you would pay progressively over time. But my mother, given the budget, had to buy this on the installment plan. And I think she was concerned that my father would be very upset by this, but fortunately he agreed that this could be an exception to his fiscal policy. It wasn't a sequester. So that sort of reflects on the facts of my parents being very interested in my education growing up.
So did that set of books, and particularly the science within it, inspire you to want to pursue medicine, or was the inspiration to go for your MD derived elsewhere?
Well, it certainly interested me in science. I had a good chemistry teacher in high school who got me excited about atoms and molecules, and I think those drove me toward thinking about medicine, and as one way where you could bring science together on health of people, and I think those are things that moved me toward medicine progressively as I went on through college.
So after medical school at Wake Forest, you ended up at Cornell to do your residency.
Right.
So how does a proud Southerner, as I've heard one of your colleagues call you, end up in New York City?
I, in medical school, had the good fortune of working with a biochemist in the field of phospholipids.
Camillo Ardem.
Camillo Ardem. And Dr. Ardem had come to this country because of the World War from Italy, where he'd been a pioneer in the field of phospholipid research. I had earlier started out interested in potassium in the heart, and I went to my cardiology professor and asked him, could I work in my free time in his laboratory, and being very honest, he said, well, I don't do anything on potassium in the heart. Why don't you talk to the chair of biochemistry, Dr. Artem? So I went to Dr. Artem with the idea that I'd like to work in his laboratory on potassium in the heart, and he says, John, that's very interesting, but why don't you work on phospholipids? So, being in the 1950s, I said, yes, sir, and that got me interested in phospholipids. And the chair of medicine at New York Hospital Cornell was David Barr, who was quite interested in HDL and its link to coronary disease. And I think those interests brought me together with Cornell.
So while you were there, did you continue your research?
Well, not as a resident. I had some conversations with people at the Rockefeller who were working in fatty acids, and but I really only got into research when I got to the NIH.
So how did you end up at the NIH?
Well, NIH was fairly new at that time, and one day at the lunch table where the residents gathered, Richard Silver, who was one of my residents, was telling us about his time at the NIH, and I said, Dick, what's the NIH? And he told me that he had been able to serve in the doctor draft at the NIH instead of joining the military. So I thought that was a good idea, and my chairman, David Barr, managed to arrange for me to get into the NIH program, the Heart Institute program.
When you were there, how did you choose which lab to work in? Or how was it that you were put in the Udenfriend lab?
We had a choice of which lab we could go to, and went around and did interviews. And I initially was interested in the lipid field and talked to Donald Fredrickson, told him I wanted to have training that would link biochemistry with clinical research in lipids. And he said, well, we're not doing any clinical research in the lab, so I think this is not the place you want to be. But then I spoke with the people in Al Scherdsman's laboratory, and they were all having a good time. It was a very productive lab, exciting things going on, and he and Sidney Udenfriend had a collaboration together that brought Udenfriend's biochemistry to bear on the clinical research in aromatic amines. And the excitement of that laboratory drew me there, and, fortunately.
So do you mark your interest in clinical pharmacology to that time?
Very much so. They were doing translational research at that time related to drugs. We didn't know what clinical pharmacology was, but that's what we were doing.
Did you ever encounter a patient while you were there, or perhaps during your residency, that shaped your approach or your interests?
Well, I think at the Heart Institute, our group managed the hypertension service. So I think it was the fact that we were involved in hypertension, treatment of hypertensive patients, that focused our research, clinical research on hypertension, and the studies that we did on aromatic amine metabolism were all in hypertensive patients because of the general philosophy that what we learned about these compounds might be relevant to hypertension.
So can you tell me a little bit about how you came across methyldopa in hypertension?
I was working in the laboratory in Sidney Udenfriend's laboratory space, and he was interested in how we could better analyze aromatic amine metabolism in humans. And we set up analytical methods based on his advances in fluorimetry to measure various amines in the urine of humans, and had perfected that as a way of of studying the effects of drugs like the monoamine oxidase inhibitors to increase the amine excretion in patients. And Sidney Udenfriend had recently been at a conference on the biosynthesis of catecholamines, international conference, and got together there with Carl Pfister from Merck and discovered that they had synthesized in Pfister's group this alpha-methyl amino acid methyldopa as part of a cancer research screen.
So it was initially targeted to be screened for cancer.
That's right.
Okay.
The Merck scientists studied it in experimental animals and had absolutely no effect on blood pressure. The classical comment from one of the pharmacologists there was, we gave a gram per kilo of this drug intravenously to a rabbit and nothing happened, so it couldn't possibly be a drug. So Sidney Udenfriend and Pfister together decided to, if Merck wouldn't do the toxicology studies, we would do investigations in patients to find its effect on aromatic amine biosynthesis in humans. And the initial plan was to study it in patients with pheochromocytoma and carcinoid syndrome to evaluate effects on catecholamine and serotonin biosynthesis. But at the time, none of those patients were in the hospital when the drug arrived, and our chief, Al Schertzma, was on a sabbatical in Sweden. So we decided, well, why don't we study these models that we'd set up on amine metabolism in patients and ascertain whether it would block their biosynthesis. And we very carefully dose ranged it up until we got to a point where we saw an effect of the drug. Yes, we were talking about our first studies on,
yes.
on methyldopa.
Yes.
We had very carefully dose ranged the drug till we got to a point where we were inhibiting the biosynthesis of tryptamine and tyramine in the patients. And looking at the patient charts, blood pressure obviously swings up and down during the day, but we had the impression that they were coming down over several days' treatment. So that night I went back to the lab and plotted out these data, which showed, in fact, that we were getting a very nice reduction in blood pressure in that first patient. And it was a very exciting graph, and I called my friend Louis Gillespie, who was working with me, who was at home, and he came into the lab, and we looked over this data and decided that this is something we needed to pursue with additional studies. So that was a eureka moment in the methyldopa story.
And I imagine it must have been rather intoxicating, as some of the first success that you had.
It was very intoxicating, and I think that's the kind of experience that drives people to continue in research, because you know that if you're fortunate, that can happen again.
Now, you moved in 1963 to Vanderbilt.
Yes.
What made you leave what sounds like a sort of Shangri-La for research and discovery at the National Heart Institute?
The clinical associates, of which I was one, typically are there for 3 or 4 years and then move out as the chief and his clinical associates. So it was time for me to migrate out of the NIH, and fortunately Alan Bass at Vanderbilt knew the work that was going on in the Schertzma Udenfriend group and contacted him, and that led to my going to Vanderbilt.
Where you established the first department of clinical pharmacology.
It was a division of clinical pharmacology in the departments of medicine and pharmacology. So we were very careful not to set ourselves up as something separate.
Not only did you do that, but you moved on to become the chairman of Department of Medicine for 14 years. So what has kept you at Vanderbilt all of these years?
The things that attracted me there were the things that kept me there. It was an institution where the pharmacology department was really committed to having a part of that department that was oriented toward human pharmacology, that would be part of the core mission of the department, to have not only their reductionist science, but clinical investigation as part of pharmacology. And the Department of Medicine, chaired by David Rogers at the time, whom I knew from Cornell, was also committed to this sort of interdisciplinary science. And then we had one of the first clinical research centers in the country was at Vanderbilt. And that was sort of a microcosm of the clinical center at NIH. So that gave us an infrastructure for doing our clinical investigation. So those are the things that drew me there, and as our program grew with a number of collaborations in the pharmacology department, it was a very productive place for me to work.
And the trainees of yours that I've spoken to have also mentioned that you protected a lot of them by being the one that raised the money and the grants and the funds in order to allow them to do their research and allow the program to grow.
I was very fortunate to have great fellows who are responsible for the work that is attributable to our program, and I do think your protected time is crucial in the development and evolution of young scientists, and that was a high priority to give them protected time and opportunity to work in this environment.
Well, beyond that, you were the translational researcher before that term became fashionable. I mean, the bulk of what you were establishing was a program to study humans and do clinical research. I mean, at the time, was that really so radical?
It was not totally radical. A lot of what was going on at the NIH was oriented in that direction, but it was a shift at that time toward translational research, and that too was fun to be part of that. Certainly, much of our research has been driven by findings in patients.
Now, speaking of your research and your personal research, much of your focus was on the biology, metabolism, and synthesis of eicosanoids. And during the course of your research, you also spent some time on sabbatical with Bengt Samuelsson at the Karolinska, who later went on to win a Nobel Prize for the discovery of thromboxane A2 and other members of the prostaglandin interarachidonic family. How did you get interested from going from phospholipids into eicosanoids?
Well, they're certainly the eicosanoids are derived from phospholipids, and but having been interested in hypertension and in the vasoactive compounds that affect blood pressure, that drew me into the area of the prostaglandins, which were extremely potent. I asked Sune Bergström, who was one of the discoverers of the prostaglandins, why he invested so much in elucidating their structures. He said it was because when you purify them down to a point in a flask that you could no longer see anything, there was still extremely potent biological activity. So that was intriguing. And in order to address this in patients, initially it appeared difficult because you couldn't measure the levels in blood because it was so small, and the drugs and the prostaglandins were cleared by the lung so rapidly. So we became interested in measuring prostaglandin biosynthesis by assessing their metabolites, which would not be interfered with in artifacts of blood collection. And I had, when I was with Judenfriend, he told me many stories about his experiences with his mentors, and one of whom had developed the first isotope dilution assay that became important in steroid, in studying steroid metabolism in humans. And we had a mass spectrometer at Vanderbilt that it occurred to me that we could apply this isotope dilution technology to measuring prostaglandin metabolites using stable isotopes instead of radioactive isotopes. So we had already developed assays with stable isotopic methods for prostaglandin E2 itself in human urine and showed that it derived from the kidney. But we would like to learn about the prostaglandin biosynthesis in the body as a whole, not just the kidney. So that led me to spend time with Mick Samuelson and learn how to biosynthesize the metabolite, stable isotopic metabolites that we'd use for mass spectrometric analysis. And once I came back from Stockholm, we invested in setting up the analysis for the PGE2 metabolite, which he already had done work on. And it was that allowed us to make the observation with my colleague John Potts that prostaglandin E biosynthesis was hugely increased in patients with lung cancer.
We were saying before the interview even started, this was, alas, unfortunately before these very easy ELISA kits from Cayman Chemicals came along to be able to measure these metabolites. Mm-hmm. So that must have been technologically much more complicated than what we're able to do now.
Well, it was a challenge at the time, but it was highly effective and specific, and I think in the immunoassay field, there was a lot of trial and error before you got an antibody that had some similarity of specificity.
Now, I know it's difficult, and it's sort of like choosing among your children, but do you have any particular favorite pieces of research that you did?
In the prostaglandin field, I think one of the areas that was important was the approach of understanding the metabolic fate of prostaglandins in order to learn their metabolites, from which we could then develop analytical approaches to measure. So our first work in that was elucidating the metabolic fate of thromboxane A2 in humans, which allowed us to identify metabolites that we could measure. And then later, we elucidated the metabolic pathway of PGD2, which allowed us to make the discovery that PGD2 is a major product of the human mast cell, and that it's released in the asthmatic airway following antigen challenge.
For which there's significant efforts for drug discovery for asthma.
Those are still going on. To my knowledge, we don't have a good DP2 antagonist in the human yet to really test its function.
One of the other major things that you're known for in terms of your research accomplishments was first-pass metabolism, discovering, it seems strange today that somebody had to discover the fact that the liver metabolizes the drugs, and that the concentration you put in is not the concentration you see in the blood. How did you come to that?
We had undertaken a study with Ciba-Geigy at the time. They had an antiarrhythmic drug, and we were interested in cardiovascular drugs. And they were having some trouble in the development because patients appeared to be getting different responses to the same dose. So we agreed to investigate its metabolic fate and pharmacokinetics if they would synthesize a tracer, radioactive tracer for us. So we gave the drug both orally and intravenously to the same individuals. And the pharmacokinetics of that time was based on what they call mammillary models, where you'd have compartments and arrows of rate constants going between them. And there was nothing there about what happened before the drug got into the systemic circulation. Well, from these studies with this no longer existent antiarrhythmic drug showed us that because we could measure the radioactivity and the in the urine and the feces that when you gave the drug by mouth, all of the drug was absorbed, but the levels in plasma were a tiny fraction of what occurred when you gave it intravenously, if you looked at the area under the curve. And that was something that wasn't built into the previous pharmacokinetic models, and led us to come up with a concept that there was a first-pass metabolism involving metabolism of the gut and the liver before the drug reached a systemic circulation. And then when David Shand in our group and Grant Wilkinson went on to use that ability to determine extraction of drugs by the liver by assessing the first pass, to developing the concept of intrinsic clearance, which is one of the fundamental concepts in pharmacokinetics. But it all started with these studies on an antiarrhythmic drug, and it was reproduced in our studies on propranolol in humans, where we found the same phenomenon.
And did this lead also to finding that there's vast interindividual differences in the way that humans process drugs?
Well, the first-pass metabolism sort of amplifies those differences, and we came up with the general concept that if you have very high first-pass metabolism, you're likely to have great interindividual variation in plasma levels. And that was the case with this antiarrhythmic drug, there was such wide differences in the plasma levels that were achieved that it was clear you couldn't use this drug with a low therapeutic index for treating patients with serious arrhythmias.
When I was looking over your CV and doing an internet search on your background, I found your testimony to a subsection of the U.S. House of Representatives on smoking education and cessation and cardiovascular risk. How did you get involved with that, and what was that experience like?
My clinical work was in the field of hypertension, and these are patients who really develop serious vascular disease, and many of our most severely hypertensive patients were smokers, in part because that is a cause of renovascular hypertension. And so I was very concerned that when I would talk to these patients, I started smoking when I was 12 years old. And
you were from North Carolina.
Yes. I even worked in the tobacco market when I was in college during the summers. So with my concern for that led me to volunteer to work with the American Heart Association, where I was chair of their smoking committee. When the American Heart began to get involved with interactions with Congress, they allowed me to be their representative and spokesperson for those hearings. And it was an interesting time because I think during those hearings, the tobacco industry and their favorite Congress people still were trying to say there was no problem with smoking and ill health. But I think when these hearings were finished, nobody ever said that again.
It seems like we take it for granted now as a known fact. Right. Now, your trainees have told me that your approach to mentoring is a bit Darwinian. Not dictatorial, but not necessarily democratic, that the people with real talent and drive absolutely excelled under your watch. So what was your approach towards mentoring trainees?
I think one important thing was to develop an environment in which people could work and succeed, where the tools were there and the ideas were there. And the other was give them protected time and to try to involve them in interesting research problems. But I think the people who succeeded were those who had an independent approach to science and were able to take problems to successful conclusions. And I was very fortunate to have a wonderful group of fellows who were very productive while they were with me.
Well, they all love you. Now, when I've talked to some of these former colleagues and trainees of yours that passed through your lab and your department, they all spoke of your love of sailing and photography. So how have you had time for developing such skills? Well, it seems like you've been also a very dedicated clinician and scientist.
I was sailing as a boy, and then in my early years in medicine, I was really too busy to get involved in that. But I guess maybe 12 years ago, a friend and I bought a sailboat, and then more recently I've gotten one of my own and have enjoyed sailing with it. We've been all the way down the East Coast from Nova Scotia down to Beaufort, North Carolina, where the boat is. But I also enjoyed chartering all over the world. I love to sail in Sweden after our time that we spent there, and the Greek islands. So it's a great pastime in which you can totally separate it from thinking about anything else.
So if you were to choose a different vocation, would it be a sailor?
I don't think so.
If you had to do it all over again, would you have chosen to be a medical doctor?
I think those of us who are involved in medicine research are very fortunate to be able to be doing what we're involved in and enjoying it.
Excellent. Well, thank you so much for joining us today and sharing a little of your story with us.
Thank you.